For our project, we chose the topic of biotechnology and cloning. So what is biotechnology? Biotechnology is the use of artificial methods to modify the genetic material of living organisms or cells to produce novel compounds or to perform new functions. Some examples of biotechnology include modifying genetic material of plants to increase their survival rate, GMOs such as crops, and gene editing and modification that is used in the COVID-19 vaccination. To understand cloning, first we need to know what DNA is. So what is DNA? DNA is a self-replicating material that is present in all living organisms and is a carrier of genetic information. So what is cloning? Cloning is the process of producing individual organisms with either identical or virtually identical DNA, either by natural or artificial means. In nature, some organisms produce clones through asexual reproduction. So how do you go about cloning? Well, there are a few processes that DNA will need to undergo before it can be cloned. These processes are gel electrophoresis and the polymerase chain reaction. So what is gel electrophoresis? Gel electrophoresis is a technique used to separate charged molecules on the basis of size and charge, in our case, DNA. The nucleic acids can be separated as whole chromosomes or as fragments. Now that we have extracted the DNA, what do we do with it? Well, the extracted DNA will need to go through DNA analysis, but before that, it needs to undergo the polymerase chain reaction. So, what is the polymerase chain reaction? The polymerase chain reaction is a technique used to rapidly increase the number of copies of specific regions of DNA for further analysis. Polymerase chain reaction is used in many real-world scenarios, such as to identify the owner of a DNA sample left at a crime scene, paternity analysis, and the comparison of small amounts of ancient DNA with modern organisms. So now that we've extracted the DNA, now what do we do with it? So, now that we have it ready for DNA, we can make clones in two different forms. We can do either molecular cloning or reproductive cloning. So, what is molecular cloning? It is when a gene sequence is isolated from a target species, synthesized, and inserted into a plasmid vector. Then that plasmid is inserted into a microbe, which is allowed to replicate and thus create copies of the originally inserted plasmid with each new microbe. An example of molecular cloning is when an insulin gene sequence is isolated from a human genome, synthesized, and inserted into a plasmid vector. Then that plasmid is inserted into an E. coli, which is allowed to replicate and thus create copies of the originally inserted plasmid with each new E. coli. These E. coli are left in a fermentation tank and left to produce insulin, which then can be collected and given to diabetics. So what is reproductive cloning? Well, it is when it's a method used to make a clone or an identical copy of an entire multicellular organism. It involves removing the nucleus of an egg cell and using the somatic body cell with its nucleus that contains the desired traits. They are then fused together to create the desired cloned offspring through a surrogate mother. Some examples of reproductive cloning include the first cloned agricultural animal, Dolly, a sheep who was born in 1996. After Dolly's cloning, several species of animals such as horses, bulls, and goats have been successfully cloned. So now that we know what biotechnology and cloning is, we're going to talk about how modern biology has helped us better understand the world around us. So as everyone already knows, every living organism is composed of cells, and these cells are the reasons for our existence. But modern biology has helped us understand exactly how these cells are created, how they function, and how cells work together to create organisms. Also, modern biology has helped us better understand genetics, DNA and RNA coding, as well as how genes are passed down to each generation. But most importantly, Modern Biology 1 has helped us develop an overall deeper and more meaningful understanding to life itself. Thank you so much for watching.